a tick, talk to the tune of a hickory stick. You were my queen in calico, I was your bashful barefoot beau, and you wrote on my slate, I love you, Joe, when we were a couple of kids.
It is wonderful to have you all back again. Keep up the good work. Let your passion for learning be the driving force for every experiment to follow. The next cell you will be viewing is found in each and every one of us. In this case, you are the human specimen. Isn't that special? Now we are ready to state the purpose of this experiment. The purpose is to visualize a human cheek cell and compare it to the cells found in the onion membrane. The materials needed for this experiment is, as you see here, a bottle of methylene blue, a toothpick, a slide, a glass slide, and a cover slip. This slide has already been prepared for you. The procedure consists of first taking the toothpick and open your mouth and rub the toothpick against your inner cheek. That's where the cheek cells are. Do not rub it too hard. You don't want to cause your cheek to bleed or anything, but rub it with a little bit of force. Then you're to collect the cells on this toothpick and put them in the middle of the slide like shown here with a drop of methylene blue and the cover slip. As you can see, there are bubbles in the specimen. These are air bubbles and they're surrounded by a black circle. You can identify them, they're all over the place. But you will find patches of cheek cells, and you need to find one that you can isolate because some of them are on top of each other because of the nature of collecting the sample. Now you're ready to look at the specimen through your 4x objective, which is a total of 40x. You're to move the cells up and down, find a cluster of cells. A lot of the cells will be overlapping, but you find an isolated cell that is squamous in appearance, slightly oval. Then you're to move to final adjustment at 100x. And you just move it very slightly. Okay, I have a good sample here. You might have to look for the cell there's going to be lots of cells that are going to be overlapping, like I said before. So you want an individual one cell, unicellular. This is one cell only. And there are many shapes to human cells versus in plant cells. There's just a few shapes that the rigid cell wall makes. You are now ready to report your observations. Take out your biology notebook. This is labeled as Session 4 Lab, the human cheek cell. The cheek cell is unicellular, one cell only. Select one of your cheek cells that look like the one I have in the picture. Using the onion cell, compare it to the human cheek cell. Let me remind you what an epithelial onion cell membrane looks like. As you can see here, there's a nuclei in all the cells. There's a cell membrane or a plasma membrane. There is a cell wall. And now you can see the human cell. The human cheek cell has a cell membrane and nucleus in the center of the cell that you can see. You cannot see the nuclear membrane. Human cells come in all shapes. This shape is squamous, while plant cells come in a few shapes. Notice the arrows. Each one is pointing to a different organelle. The yellow arrow is pointing to the nucleus. The black arrow is pointing to the cytoplasm. And the red arrow is pointing to the cell membrane. Now you are ready to draw your cheek cell. Label the cheek cell and include the cell membrane, the nucleus, and the cytoplasm, and the nuclear membrane. The cell membrane protects the cell from its external environment. One of the functions of the nucleus is to contain the chromosomes, which is where our hereditary material is stored. You cannot see the nuclear membrane, but dot it in. The function of the nuclear membrane is to contain the chromosomes or the protein. Then you see the cytoplasm. Shade in the cytoplasm. This is all the material found outside of the nuclear membrane. It's a thick solution that fills each cell, mainly composed of water, salt, and proteins. There is a small vacuole in the cell. This small vacuole keeps the nucleus in the center. You cannot see the vacuole, but dot it in. Label each cheek cell organelle and put the function next to it. Remember that in your notebook, you should include first the purpose in the introduction, then the materials, then the procedures, and now we are completing the observation. You are now ready to draw and compare the two cells, the cheek cell versus the onion cell. In the cheek cell, we pointed out 5 organelles: the nucleus, the nuclear membrane, the plasma membrane or the cell membrane, the small vacuole. We used the dot system to draw in the nuclear membrane, the plasma membrane or cell membrane, the small vacuole, in the onion cell, we pointed out 6 organelles: the nucleus, the nuclear membrane, the plasma membrane, the large vacuole, and the chloroplast. Use the dot system to identify the nuclear membrane, the plasma membrane, the large vacuole, and the chloroplast. You are now ready to finish biology lab number 4. Write your conclusion and put your microscope away. First, remove your slide. The objective has been moved from 10x, which is 100 total magnification, to 4x, which is 40. That's how you're going to store the microscope. Then take a piece of lens paper and clean off any liquid or dust. Clean the objectives well and the ocular lens. Now look at the stage. The stage has some dust on it. Remove it. Now you're ready to turn off the light. And we're going to put the cover on. And voila, we finished another lab. See you later.